You are in a very special place. This theater has provided entertainment to the Hudson Valley for over 100 years, from vaudeville to the USO to the latest independent films. Lovingly run by the Caccio family for over 60 years, for the last 10, the theater has been operated as a volunteer-run nonprofit and one of the few small-town single-screen theaters left in New York State. Bringing you incredible films, theater, and music events is what we do, and we want to do more of it. Help us build the next 10 years of Rosendale Theater by dropping a little something in the donation box on your way out. It makes a big difference. Now, enjoy the show.
Hi, everybody. Here I am at the Rosendale Theater. Just look behind me. This is what we've all been missing. So I just want to reassure you that we've been hard at work behind the scenes. We are planning on a very rigorous cleaning protocol. We will have social distancing, limiting sizes of audiences, and following every rule to keep you safe and healthy. In the meantime, you go to our website at rosendaletheater.org, and you can see all the virtual streaming we've done. We've done a few live events that you can't see anywhere else. And tonight, you will see a part of our Science on Screen, a panel that was set to happen here in the theater live, and now we're doing it virtually. So tune in. Oh, you are tuned in, and enjoy the panel. Thanks.
One.
Hello, and welcome to the Rosendale Theater and our second online discussion as part of our Science on Screen series. Tonight's discussion on viruses, what they are and how they spread, was going to be in the theater live after the screening of the film Contagion. We are still live, but tonight we're online. The fictional thriller about a pandemic has been left behind by the actual COVID-19 novel coronavirus pandemic that shut our theater and much of our activity down everywhere, at least temporarily. You may have already watched the movie and can see Contagion online through Amazon Prime. We were not able to screen it through our portal, but we know that tonight's discussion will prove to be quite a bit better and on point for thinking about what is going on and may provide some answers to many of our questions. Tonight's panel, our biologist-virologists with us are Dr. David Esteban, Associate Professor of Biology and the Pauline Newman Director of Science, Technology, and Society at Vassar College, and Dr. Maureen Morrow, Professor of Biology at SUNY New Paltz. I'm Pam Cray, I'm your host from the theater. Our moderator for the panel tonight, thank you, Jim Metzner, host of Pulse of the Planet radio series, is going to take it away, and I will disappear. Thank you all, and thank you, Jim. Here you go.
Thank you, Pam, before you disappear. I just wanted to reiterate how kind of weird it was to be involved in this thing, months before anybody was even thinking about pandemic, and making this choice and seeing what happened. You can't help feel cause and effect, even though that's magical thinking. And all I can say now is the good news is the movie we chose didn't have any zombies in it. Well, for those of us who had heard or not, I'm just going to say, pointing out that there's a group of people who deserve our gratitude and thanks because they're on the front lines of research. And as you're about to hear from tonight, one of the mantras that you'll hear tonight is that there's a lot we don't know. And we'll probably say that a lot. But interestingly enough, that's what inspires scientists to go further, to discover. The playing field of science is the unknown. Scientists are heroes. Cue the picture, please. This is, or should be, a photograph of June Almeida, who discovered the coronavirus in 1964. Thank you. And I just wanted to say that, pardon me, just wanted to say that if there's any students watching tonight, if you're looking for a career that's really challenging and helpful to all of us, then consider being a scientist. And we will all now and in the future be glad that you did. Who knows what discoveries you may make. So I'm going to turn this over now to our two doctor professors who are going to introduce make a brief introductory statement, starting with Dr. Maureen Morrow.
Thanks, Jim. I just thought it was worth commenting on the fact that these virologists, scientists, we get kind of excited when there's a pandemic, and that seems kind of mean because we recognize that this virus has disrupted lives and livelihoods, and many people are suffering. So keep in mind that if we get a bit excited about what we're talking about, both David and I have great compassion for the ways that people are suffering, and great respect for the people who are dealing directly with COVID patients and those other essential workers that are dealing with those of us that are showing no symptoms, but maybe being exposed through asymptomatic carriers. We really do appreciate what people are doing. So forgive us for getting really excited about some of these topics. Another underappreciated group that you alluded to there, Jim, is researchers who do basic research. So there are people who have been studying coronaviruses for decades, and for most of that time, coronaviruses were boring. All they did was cause the common cold. Who cared? Hard to get funding for that work, but thankfully there are people who are passionate about those viruses, and their work has set us up to be able to deal with this virus much more quickly than we otherwise would have. So let's all, three cheers for the basic research scientists also. So with that little bit of a point, I'll turn it over to David.
Thanks, Maureen. So I just wanted to start off with saying a few things about how this started and how that fits into what we've seen before. And so as we think about, where we were several months ago with this new virus suddenly appearing, where do new viruses actually come from? And so this pattern we've seen before. New viruses come from animals, and as we encroach on animal habitat, as we interact with animals more and more, viruses that are naturally circulating in animals have the opportunity to infect humans. And most of the time when that happens, nothing happens. The virus does not successfully infect the human, or it's not able to continue the transmission from human to human. But occasionally it does succeed. And that's happened over and over. So it happens, that's how Ebola outbreaks start. That was the start of the Zika pandemic, the first SARS epidemic. Smallpox thousands of years ago spilled over from animals. So this happens repeatedly, and it'll keep happening in the future. This particular coronavirus, its closest relative is in bats, and so at some point it spilled over from bats, perhaps into another animal. We're looking at a picture of the coronavirus here, and from there infected humans, likely through some exposure to bat guano, for example. And it's followed exactly the pattern that we would expect from these kinds of spillover events. And from the perspective of this particular virus, this one's been very successful in spreading human to human. whereas others are not necessarily quite this successful.
Well, thank you, David. We're going to go to the questions and answers. We have some questions first, and then I'd invite those who are listening, if there's a question that comes to mind, particularly early on, we'll be scrolling this. I think maybe you're already seeing it, but I think you can send them either on, if not on Facebook, send it to the Rosendale Theater. Theater is spelled with an R-E at the end, not E-R. The Rosendale Theater at gmail.com or on Facebook, and we'll get those questions. But we have some from starters, and a lot of people have sent in questions already. So here we go. The first question is sort of following on what David was just saying. Let's get a little background here. What is a virus? I mean, I've heard that they're kind of not alive, and how could that be? How do they operate? And what's a virus?
So let's start with the question of whether or not they're alive. If you follow the strict definition of what you'd see in a biology textbook, then no, they don't meet the criteria for being alive. But they clearly have a role in biology. They're not cells, so all other organisms are made of cells, and viruses are not. But they do depend on cells to replicate. They're basically a piece of genetic material. In the case of coronavirus, it's RNA, which is a lot like DNA. Same basic idea, but it's a slightly different molecule than DNA. So it has this RNA as its genetic material that's wrapped in a protein coat that helps protect it. And then that is wrapped in basically like a membrane that surrounds it. And in that membrane, there's these little spikes. And you've probably seen these images, and that was the electron microscope image that we just put up a moment ago. You could see these little spikes coming off of the surface, and that's where it gets its name, coronavirus, because it looks a little bit like a crown. Those spikes are really important for the virus because, as I mentioned, they need to infect a cell to be able to replicate. And that spike, what it does is attaches to the surface of the cell that it's going to infect, and then starts that process of getting inside the cell. Once it's inside, it basically takes over the processes in the cell to use the cell's own machinery, its own resources, to actually start making new copies of the virus. So it's totally dependent on that cell, but it really comes in and exploits the processes and the resources of the cell, makes dozens, hundreds, thousands of copies of itself, and then those virus particles are then released back into the environment, where they'll infect the next cells in that particular host individual in that tissue, or perhaps transmitted to the next person.
Thank you. It's amazing to wrap your mind around something that isn't alive, but still has this imperative to reproduce itself by infecting us. And the only way apparently it can do that is by commandeering, by being like the ultimate invader, I guess. So we'll keep in mind what you said when we come back later to questions about drugs and so forth, and how, and vaccines, and how they might help us in defeating the virus in the way it operates. So how is COVID-19, and I don't know, maybe you want to talk about the name as well, but how is this virus, this is the famous Passover question, why is this virus different from all other viruses? How is it? Is it in fact different from all other viruses? And if so, how?
Yeah, so first, to clarify the terminology, the virus is actually called SARS coronavirus 2. And COVID-19 is the disease that it causes, sort of like HIV is the virus and AIDS is the disease. So how is it different? Actually, maybe I'll take a slightly different spin on it and talk about how is it the same. And so if you compare it to SARS-1, its genetic material is about 80% identical. If you compare it to all other viruses, basically all viruses have the same set of problems that they need to overcome. And you can actually simplify a viral replication cycle into key steps, and all viruses have to do those steps. But where it's really interesting is when you get into the details, because each virus solves those different problems in slightly different ways. And it's amazing sort of the diversity of solutions that have evolved to all of the different problems that viruses have to overcome. So in that way, they're all similar. They follow that same pattern, but the details is where they're different and especially interesting. So if we look at SARS-1 and SARS-2, there's some important differences. So that spike protein that's on the surface that I mentioned is so important for attaching to get into the infected cell. It's a little bit different from the one in SARS-1, and it's possible that some of those differences are, there's the spikes there you can see on the surface in this electron microscope image. It's very similar to the one in SARS-1, but there's some slight differences, and it's possible that those differences might contribute to its ability to transmit and infect more effectively than the first SARS virus did.
Okay. Then just quickly a bit of history. SARS-1, SARS-2, when did we first encounter SARS-1?
SARS-1 was 2002, 2003. Started in a very similar fashion, bats to a couple of potential other animals, and then to humans. But the number of cases was much smaller than the current pandemic, although the mortality rate was much higher.
At some point, we're going to talk about mutation. I think we might have mentioned it, but that plays an important role too. I don't know if someone wants to get into that now or later, but I think it's something we want to talk about, right?
We'll definitely talk about it. We can save it for later.
Okay, mutation, just hang in there. All right, so
I think one thing that's really interesting to consider here also with viruses, whether they're alive or dead or whatever, that this SARS virus has 29 genes and humans have a bit less than 29,000 genes. So it's just amazing that these things exist with just the tiniest amount of genetic material.
Yeah, indeed. And is there, you talked about similarities. Is there anything significantly different? I wanted to just circle back to the question, why is it different from other viruses? Is there anything about this particular virus that sort of separates it a little bit from the pack? Is it the spikes? I mean, what is it? Is there one thing or one category of things?
I think it's one of those questions where it's in the details. And at this point, I would say we don't know exactly what's giving this virus its slightly different properties from its closest relatives. There isn't there isn't like a whole gene that's completely different or big. It's a bunch of little things, and how those little differences add up. We still don't know what each of those differences contributes to overall what we're looking at.
So if you had to pick maybe one or two significant things that we do know about it, I mean, there's a lot we do know, and there's something we know about it, and there's something we don't know. So just a little bit of both. What do we know that's significant, and what do we not know that's big time significant? What's like, wow, if we only knew that.
Yeah. So what is it that we do know? I would say, I think this difference in the spike is really interesting, in that if you compare it to SARS, so the spike actually needs to be modified slightly for it to function, and that's done by an enzyme, so that just cuts it and changes its shape a little bit, and that's necessary for it to function. In SARS, that happens right at the beginning of the infection, and in this coronavirus, SARS coronavirus 2, it probably happens at the end as the virus is leaving the infected cell. And it's possible then that because it's already modified and ready to infect, that's what makes it more infectious. That's a hypothesis, but it's an interesting finding about that difference. Things we don't know, I think there's a couple of really important questions. One of these I know Maureen will address is the issue of immunity. The other one I want to mention that I think is really important is long-term effects. We kind of get this message that it takes a couple of weeks to recover, but we're seeing cases of people that take a lot longer to recover. And of course, we don't have the long-term view yet, but are we going to see long-term chronic health issues related to coronavirus infection? I think that'll be a really important question to address.
It's just the interesting thing about this is, like, I feel like we're all in the Petri dish. I feel like we're all participants in this experiment. It's not like we're talking about this distant scientific experiment. No, this affects all of us. You're talking about stuff that this is science that's now come home to everybody. Everybody on the planet needs to know the answers to these questions. So, Maureen, immunity, such a big issue. Why is it that some people get apparently mildly ill and others get seriously ill, seriously enough to die? What do we know about that, this issue of immunity?
Not a whole lot. It's clear that there are comorbidities that increase the likelihood of having a very severe reaction to this virus. Comorbidities, other illnesses, and sometimes just even polluted environment that makes a person more likely to have this severe reaction. What I don't think a lot of people appreciate is how complicated the immune system is. I think we're all comfortable with the idea that our brain is very complicated. There's many interactions, many different types of cells, and we accept that. But our immune system is composed of many different types of cells doing different jobs, communicating with each other, and the immune system has memory, sort of like the brain also. And the point there is the complications, or the complicated nature of the immune system makes it so that we really don't know everything about it. And importantly, the immune system reacts when a foreign object enters the body, like a virus or a piece of pollen or something along those lines, and the immune system revs up, does its thing, and then normally it shuts back down. And it seems in these certain situations, a small number, or small percentage, I shouldn't say small number, small percentage of people, their immune system just keeps going. And part of the immune system reaction is to create cytokines, and those help stop the growth of viruses. When that doesn't tamp down as it should, you get this cytokine storm, is causing all the respiratory problems. And then, of course, we'll address it later, there are other stages that many people go through where there's increased clotting and then other hyperinflammatory reactions. So the short answer We don't know. The medium answer, we don't know because the immune system is so complicated.
So I wanted to ask you about just another example that we're all familiar with is like allergies. Like I'm allergic to something and you're not, and we're both walking through a field and I start coughing and sneezing because I'm allergic and you don't because you're not allergic. Is that an immune system thing? Is this an example of what we're talking about?
Yeah, so the immune system's complicated and it can kind of go awry for various reasons. But there's also genetic differences. We have great diversity in our immune system. That's why we can't easily exchange organs. You know, organ transplants are a big deal because if you take in someone else's kidney, your immune system is going to recognize that as foreign, as not being normally part of your body. And the diversity in our genes and the diversity within the complexity within the immune system leads to these kinds of differences.
So this is one of the big questions that we don't know that we really want to know. Why this question of why certain people die and other people, well, there are certain populations that are more at risk than others. We've heard that, right?
Mm-hmm.
I mean, I'm as a senior, as I'm a senior, I'm supposedly more vulnerable for whatever reasons, right?
Correct. Yeah, the immune system starts to slow down with age. It doesn't respond as vigorously.
That's right. As we mellow our way into seniorhood, there's another thought to cheer us up. So we'll probably come back to that. Oh, my goodness. Life enters into the picture. Sorry. That never happens. Vaccines and drugs, a segue to vaccines and drugs. How do they work? And so now our immune system is doing the best it can. It needs help. Along comes a drug or a vaccine, I suppose even better. How do they work? This is such a big one. Who's going to tackle this mighty question?
I'll tackle the drugs, and I'll let Maureen tackle the vaccines. So developing antiviral drugs is actually quite challenging, and one of the reasons, right? If you think back to they're not cells, and as Maureen pointed out, this coronavirus only has about 29 genes. When you and that it's taking over and using our own machinery to replicate. When you use a drug, it's going to be something that's very specific to blocking a specific process. Something, they're very precise in what they're trying to stop. And so because the virus uses so much of our own machinery to replicate, then the targets are actually our own. And that would therefore be something that would be toxic to our own cells. And so the only things that we can really target effectively with an antiviral drug is something that is unique to that virus. And since they're so small and are taking over our own processes, there's not much to target. But thankfully there are some antiviral drugs, and there are some that were already in development for other viruses, and they can be tested for against corona. One that's showing some promise is remdesivir, which is a drug that specifically blocks an enzyme that copies the viral genetic material. So I mentioned that it's an RNA virus instead of DNA. So it has a special enzyme that copies that RNA that cells don't have. This is a uniquely viral thing. So this drug, remdesivir, is able to block the activity of this enzyme, and so the virus can't make copies of its genetic material. Another approach, drug-based approach, that instead of targeting a viral process, is actually targeting the immune system. Can we find a drug that would calm down the immune system, that would prevent it from overreacting, so that would, perhaps help reset it. And there's some promise there as well for some immune modulating drugs, such as ones that used in like rheumatoid arthritis. But that can be complicated too, because you don't want to steer the you don't want to turn the immune system off. You want to steer it in the right direction.
Yeah. Oof. Okay, we're going to go to vaccines. Maybe Maureen will talk about that, but I want to ask both of you the question about, is there a timetable? I mean, is there a realistic timetable, both for, remdesivir exists, they're doing testing now, so maybe it's more of a vaccine question, but what is a realistic timetable? Maybe that's an introduction to the vaccine.
Okay. Well, with the remdesivir, we know it's not working perfectly, and probably with more research, we're going to realize it has to be administered pretty early in the infection process. And unfortunately, people come to the hospital when they're very, very sick. It might be too late for that drug to have a significant effect. And in terms of vaccine development, we know they're really fast tracking and allowing phase 1, phase 2 trials and phase 3 to overlap a bit. And the FDA is really stepping back a bit from their usual caution because we have to. It's quite possible that we'll have a vaccine by early next year, early 2021, but that's with everything going perfectly. So why is it so hard to come up with vaccines? What we're basically doing with a vaccination is mimicking an infection. And so we're riling up the immune system and potentially can set the immune system out of balance and cause side effects that way. A lot of the vaccines people are familiar with are the actual virus itself, and that virus has been killed or what they call attenuated, made so that it doesn't really cause infections. And those are very effective vaccines because they mimic the infection very closely. Some of the new vaccines that we're hearing about in the news are using newer technologies that will likely get rid of some of the potential negative side effects of injecting a person with a full virus, or many of them actually. But because they're new, we don't know that they won't have some kind of strange effect on our immune system. And that's why we have to go through these trials.
Sure.
And in each trial, if you read about it, a bigger and bigger number of people are involved. And the more people that are involved, the more likely you will see one of these side effects. So if the side effect happens in 1 in a million, you're not going to know until 1 million people have received the vaccine. So that's why the FDA is so careful and slows things down. But I think wanted to mention some of these new vaccines and how they are designed. And so like I said, we're trying to mimic the infection with a vaccine. And if we're not going to put the entire virus into the body, we can just put parts of the virus into our body and allow the immune system to see those parts. And if it's just a part of a virus, then you can't get the viral infection. So as David was mentioning, the spike protein is hugely important in the infection process. That spike protein is what binds to our cells, and binding is like, sticking to the cells, and it's very specific. If we have antibodies that first bind to the spike protein, then that binding site is covered up by the antibody, and the virus, the spike protein of the virus can't stick to our cells, and then the infection never starts. So we can put just plain old spike protein into a vaccine, and that will allow our immune system to see this foreign entity and create antibodies that would then neutralize the virus, prevent it from being able to bind to our cells. But some of the most promising vaccines include this mRNA vaccine. It's a company, Moderna, from Cambridge, Massachusetts, is working on this. And basically what we're doing is putting the RNA that David talked about, the thing that the virus puts into our cells, and then has our cells do all the work to make spike protein and everything else that this virus needs. So we put that mRNA of just the spike protein into our cells, and then our cells make this spike protein and share it with the immune system, so the immune system can now say, oh, we gotta take care of that and make antibodies to that spike protein. And then another version of a vaccine that they're developing is done by taking a fairly harmless virus, an adenovirus, and genetically changing it so that the adenovirus makes the coronavirus spike protein. So this, the adenovirus is called a vector. So this vector is going to carry that genetic material into our cells and carry that spike protein that the immune system will be able to respond to.
Can you remind us, you mentioned antibodies a couple of times. Antibodies they're like, I'm imagining them as sort of like the foot soldiers of our immune system, but is that too simplistic? What are antibodies in reality?
I don't know exactly what a foot soldier does, so I'm not really sure what the analogy is there. I'm sorry. But yeah, so antibodies are proteins, and they're part of the immune system. Well, so if we think like what's in our blood, we know we have red blood cells, we have white blood cells. We talk about white blood cells. People may be familiar with that. They're important in infections, and one subtype of white blood cells are called B cells. B is like bird. And those are the cells that will make these antibody proteins after they see this foreign protein in the body. And the antibodies that they make are very specific. These antibody proteins will really only bind to the spike protein from the SARS-CoV-2 and not other things.
Mm-hmm. So the antibodies could do many different things, but they have to be given instructions as to what to do. Is that basically it by the immune system?
Basically the B cells have to get the instruction. They have to see this strange protein and then say, okay, this is the antibody we got to make here.
Okay, let's move on, because I want to get to some of the questions that have been submitted by our attendees in advance. Lag time, it's sort of an issue that's come up a bit in terms of, God knows, we listen to the news and we see all the stats that we're getting. This is what's happening now, and yet what's really going down now, we won't really know, get a good picture of it until what, 3 weeks from now? I wondered if you could comment on it. It's sort of an odd thing of having, like, for example, they're letting the beaches open in certain places, but we don't really know what the effects of that will be till 3 weeks from now, for example. So what about lag time and how that affects this? And is this all lag time due to what, lack of testing? Is that it?
So I can speak to that a little bit. So it's one of the challenging features of this particular infection is the really long time between exposure and infection to when you actually start to see symptoms. And, it can be as long as 2 weeks. And then, those initial symptoms may not seem like much, especially now. You mentioned allergies, maybe you think it's allergies. And then often it's not until someone has fairly severe symptoms that they might actually get tested. So it could be 3 weeks, 4 weeks before that person is tested and diagnosed. In which case, if you want to know, all of the people that are showing up with infections were exposed, maybe 3 weeks ago. Then you have to figure out, right? Was it the open beaches? Was it dining out at a restaurant? And if you try to remember back, what you did 3 weeks ago, that can be challenging to figure out. Part of it is testing. So if we could do testing regularly and right away, we could shorten that down. But part of it is just you may not even think to get tested for 2 weeks.
Well, I meant to ask you about that first, but I'm so glad we need to get into testing a little bit, of course. So they're basically what, 2 basic kinds of tests, whether you have the disease or not, and then whether you have the antibodies, whether you've had an immune response. Is that it? Are there different kinds of tests? Anything in the pipeline for better tests? Anything at all you guys want to weigh in on testing?
Sure, I can. I took a look at what antibody tests are currently available. These are new tests, and they're approved through the FDA through something that's called emergency use approval. So much quicker than tests would normally be approved. And there are a number of different ways that you can see if antibodies are present there. We can't look under a microscope to see them. So there's these indirect methods where through different tricks and techniques, and you're able to visualize when there's an antibody that's specifically binding to the spike protein, or some of these tests focus on other proteins of the virus. And several of these tests are rapid tests, very similar to the rapid tests for influenza or pregnancy tests. They're engineered very, very similarly. But these tests have less than perfect accuracy. So the one big one that's being used has 93.8% sensitivity, so not 100%, and 95.6% specificity, not 100%. So there's false positive reactions, and then there's false negative reactions, meaning you might not have the antibodies, but the test will give you the blue band, and you might have the antibodies, but that blue band won't show up. So that's why sometimes people come up with a negative antibody test. There are, when they've actually been exposed. Some of the other tests are more complicated, they take longer. And I think one of the best tests is a newer test by Roche that is showing 100% for sensitivity.
That's an antibody test you're talking about there?
Yeah, but not the quick rapid one. It takes more time to get the results.
What about the test to see if you have the virus? The one where I think they stick a swab up your nose or something. Is that?
Yeah. So the those tests operate on a different principle. So the antibody test can tell you if you have been exposed and developed antibodies, and so that would reveal a past infection. But if you want to know if you are infected right now, the common one is the nasopharyngeal swab. So they stick it up your nose and they swab sort of the back of your nose. And what that's doing is actually collecting a sample that hopefully has the virus in it. And the principle of those tests is that they're actually detecting the viral genetic material, the RNA. And so it uses a technique to take this very, very small amount of viral RNA and make lots and lots of copies of it so that you can actually detect it. And that technique itself is actually a fairly straightforward technique to do. But of course, the key issue, as Maureen was pointing out, is the false positive and the false negatives. Those have to be, low false positives and low false negatives. But that one, all of those tests are based on some variation of detecting the genetic material, the RNA. But there's other ways to do it. So there's some being developed that are for testing saliva instead of the nasal swab, but they work on the same principle.
And I've heard, this wasn't on our list, but it's certainly coming to mind, there's so many different varied symptoms, and some of them are odd, like losing your sense of smell. I've heard that being as a possible symptom, certainly getting fever and starting coughing and all of that, but some people are, what is the word, asymptomatic. So there's a whole, the other thing that's weird about this that makes it, I think, different maybe from other viruses is that symptoms are like all over the map. Am I right in that? That's at least how it seems to me.
Yeah, clinically, it presents in so many different ways. And I'm not a clinician, so I kind of have a superficial understanding of this. But some of the thoughts are that if the virus is killing off cells in your nose, those cells necessary for detecting odors might be destroyed or compromised in some way, that you lose that sense of smell. And the blood clotting might have to do with when the virus starts killing cells in our blood vessels, and the response that follows is, okay, now we need to start clotting, so let's make a lot more of these clotting factors. And then there's all these weird things that happen after you recover from the respiratory distress, and after you pass this high clotting activity, then there are some other weird hyperinflammatory reactions that still we don't understand what is going on, whether it's the virus itself doing some kind of damage, or whether it's the immune system that's really gotten out of control.
Yeah.
And yeah, so some of this stuff is really fascinating. I know at one point I did want to mention there's this great podcast called This Week in Virology, or TWIV for short. And for anyone who can deal with maybe hearing a little bit of jargon and having some of the information go over their head, I highly recommend it. They often have this clinician come on and give reports of what is being seen in the hospital, and all these really fascinating and scary reactions to the virus.
Thank you. I wanted to, so moving on, what is the likelihood of this pandemic dying out or being eradicated?
The short answer is, I think, essentially zero. The longer answer is, I think there's enough transmission going on globally that the chain of transmission from human to human, there's enough virus and there's enough still susceptible people that chain is just going to keep going. So I think what we're going to see is not that it's going to disappear, but rather it's going to become endemic, which just means it's just going to become part of the background of infectious disease that we see every year. Hopefully not in these numbers and in this severity, but it's going to become it's something that we have to learn how to live with. In terms of so why do some viruses actually disappear? So think back to the Ebola virus epidemic in West Africa. It did eventually disappear. And this is what happens with Ebola, is each new outbreak is caused by one of these spillover events from animals into humans. There's a sustained human to human chain of transmission until there isn't. And then the next outbreak that occurs is not the same virus coming back, but it's a new strain coming back from animals. So some will just disappear because they can't maintain that chain of transmission. I don't think that's going to happen with this one. So then the other option is to do what we did with smallpox, is develop a really good vaccine, have a mass vaccination strategy, and that the vaccine is effective, and that we have high acceptance rate, high compliance of people taking the vaccine, to do it that way. But that's been done. Smallpox was eradicated. Polio is really close, but it's been really hard to get the numbers down to zero. So those are enormous accomplishments, but clearly not easy to do.
Well, this virus is, I think, related to the so-called common cold, and there is no vaccine for the common cold. Or the flu, for example. We have to reinvent the wheel because the viruses mutate and change every year. Are we in the cards for having to change a vaccine every year, or what's the prognosis for can you create a vaccine for something, for a virus that may mutate? And like the common cold, elude us because it's mutated, it's changed, and we will always be chasing it.
I think one of the things about the common cold to keep in mind is because vaccines have a low rate of adverse effects, we don't develop vaccines against viruses that aren't very deadly. And in addition to that, there's a lot of diversity in the cold viruses. But for this scenario, a vaccine really is what we're going to have to do. Unfortunately, the virus does not mutate the way influenza does. Influenza, it's a whole other story, very interesting virus that becomes a very different thing when it infects pigs and then moves into humans. And it's quite cool. But this virus, low mutation rate, and in particular, in the spike protein region, there seems to be an even lower mutation rate, and that's where our vaccines that are going to be very specific against the spike protein. That spike protein isn't going to change and no longer be able to bind to the antibodies.
So there's some good news, potential good news.
Yeah.
So how do best, we're going to get to the attendee questions in a second, but how to best prepare for the future? What's in the cards, sort of near and long term? Anybody want to weigh in on that? Peeking into the future.
Did you say you wanted to address that, David?
In our previous discussions, I liked your answer, Maureen. So why don't you go ahead?
What was my answer? We have to vaccinate. We have to continue doing research. Really, in the long-term future, some other virus is going to come along, and if we don't have the basic research into how to develop safe vaccines and how to understand what new viruses might come along, we will be in trouble. So I think we need to keep doing the research.
I think this really ties into Maureen's opening statement about the basic research. All the people out there that are researching viruses that are not the popular ones, we have to keep supporting that work because, it's, I know that it seems like it's taking a long time to develop vaccines and antiviral drugs against this virus, but we have known about this virus, for a couple of months. And the pace of scientific research is typically much slower. And the reason that we can even say something like there might be a vaccine if everything goes well, in 10, 12 months, is because of all that work that people were doing, developing a vaccine against SARS and other coronaviruses. When people said, SARS isn't around anymore, why bother developing a vaccine for it? Because they knew they knew what could happen, and so it's important to fund that kind of basic research.
Yeah, absolutely. It's so important. Um, okay. Well, any good sources of intel? Good. You mentioned that that podcast. Anything else? Any place that we can turn to for good, reliable news and the science behind this that you can recommend?
I'll second Maureen's nomination of TWIV as an excellent resource. It really is.
TWID?
TWIV, This Week in Virology.
TWIV. Okay, thank you. As a podcast, and then of course we get all these briefings that from WHO and the World Health Organization. Good source.
Listen to the scientists.
Yeah.
And the public health officials, the experts, people who have been studying this stuff for a long time and dedicated their lives to it. You know, they have a strong understanding of how things work and what we need to do.
All right, so we're going to get to some questions, some of which we've touched on. We have some great questions from folks who've mailed them in. Now, I'm going to mispronounce somebody's name. I hope not. Joanna Rotte, R-O-T-T-E, Joanna, and Deb DiPietro essentially asked the same question, which is that noting that Deb said, the way Deb puts it is, I'm curious to know if the COVID-19 structure has changed or mutated since it was discovered. What she understands, the vaccine is in trial, and she wants to understand how effective the vaccine would be for this virus might be compared to the influenza vaccine, where flu vaccine is produced using strains of virus in hopes the cocktail will provide the most coverage. Does COVID-19 seem to behave like the influenza, or is this a very specific, is there a difference in its structure whereby a vaccine produced would likely be more effective at affecting immunity?
Well, we kind of touched on this a little bit before. The SARS-CoV-2 is not mutating in the way that influenza does. So a vaccine is likely to be, you make one kind of vaccine and it will work for long times. The question at this point is, will the vaccine create a long-term immunity, or are we going to need to get revaccinated every year? There's something quirky about these SARS viruses that they seem to not induce a long-term immune response. And well, that's another exciting thing to learn about in immunology and virology is why isn't it fully activating all of the parts of the immune system that will give us this memory, this long-term ability to continue to produce antibodies to the virus.
Okay, we have a couple of questions.
And also, can I just sort of quickly address the issue of mutation and strains, because
yes,
it always sounds scary when they say things like the virus is mutating, and I just wanted to point out that mutation is just a normal and natural process that always occurs in viral replication and in cells replication as well. Viruses do it at a higher rate though. But that doesn't mean that it's necessarily taking on new properties. So we're seeing some mutation occur, but it doesn't look like any of those variations actually have differences in their properties. They don't transmit better. It doesn't cause more disease. And so there aren't really different strains at this point, but there are slight differences in the genetic sequence, and you can track those, right? That's why we know that it likely came to the US through Europe, not directly from China, because you can essentially track the genealogy of it through those mutations.
Okay, thank you. We're gonna thank you both. We're gonna move along because I want to try to get all these questions before we have to wrap up. From Wendy Saul, given the huge number of people who have been infected worldwide, why do we seem to have so little information about how it's being transmitted from one person to another? that true that we have little information how it's being transmitted?
Yeah, we certainly do have a good sense of how it's being transmitted. There are many questions about the exact details of how it's being transmitted, whether you need to be 3 feet apart or 6 feet apart, how far these aerosols move, is the virus still alive when it's moved 6 feet in through the air. These are studies that will probably be done at some point in time, but we certainly can't like bring a bunch of human volunteers into the lab and say, well, we'll spray some virus at you and see what happens. So there's still tons to be learned. And I think a lot of the recommendations that we hear are best and safe kind of guesses based on what we know, what's likely to be the best way to go.
Thank you. Oh, you answered the second part of Wendy's question too. You anticipated that. This one from Maria Reidelbach. If the virus decomposes on all surfaces after a couple of days, well, does it? I guess that's really the basis of her question. What about viruses on the surface? Do we know that? Is that something we've found out?
Yeah. So again, if you think about the structure of the virus, it has this membrane with the spike proteins in it. That's actually a fairly delicate structure. So when it lands on a surface or even in a particle in the air, that's going to break down over time. So there's some studies that look at how long can you detect the virus on, cardboard versus metal versus, plastic and so forth. And you can certainly pick up, in those laboratory controlled studies, you can detect live virus there. What we don't know is how much does picking up virus from a surface contribute to the overall transmission that we're seeing.
Yeah.
That's harder to answer. And one thing I would actually caution about when you read news articles about studies like this, there's virus on these surfaces or they found virus in the air, is to look carefully. Are they detecting viral RNA or are they detecting live virus?
Huh.
Because you can detect the RNA, but it could be that membrane is totally destroyed, and so it's not going to be infectious. If they're detecting live virus, that tells you it's still functional and could still potentially cause an infection. The presence of RNA alone, there's the potential, but we don't know yet if that could actually cause an infection.
There's the zombie. The virus is live, but it's dead. So put that in your pipe and smoke it. Anyway, moving. So it's a good idea to be sort of anal and clean up a lot and wipe down all the surface until we know more. I mean, yes, we could be getting the RNA, but why not? Until we really know better, why not be, a little bit.
Go ahead and clean it.
Go ahead and clean it. When in doubt, clean it.
And wear the mask.
And hey, maybe it won't help with SARS coronavirus too, but it'll probably help clean off all kinds of other nasty germs we want to avoid too.
Really, we may all be getting less of the common cold. There you go. Moving on from Pam Cray, the virus as it is depicted, I've seen relatively the same shape, but different colors. Can you talk about the virus as it looks under the microscope? Or is this sort of like what, we see all these fabulous, in Astronomy magazine, you see all these fabulous, day-glow colors, and then you look in the telescope and it's like, wait a minute, that doesn't look like Astronomy magazine at all. So what's going on here? Is this like, with the colors, is that added on? Are we adding food coloring to the sample? What's happening?
Yeah, well, we saw that image before. It's the black and white image that you get from an electron microscope. And we need electron microscopes because nothing else can see something that's small. The electron microscope, instead of visual light that our eyes perceive, it uses electrons to visualize these viruses. And so what you get from the electrons is some computer generated image. It's a digitized image. And then we know what you can do with digital images. You can manipulate them in any way you want and add color to different parts of the structure. But what we're seeing in a lot of the images for the SARS-CoV-2 is really, I would call it the avatar of the virus, because those are animations that graphic artists put together based on the real electron microscope image. And those are, they're fantastic looking, and they're 3-dimensional, and each protein has a cool color, and they make the viruses look kind of cuddly in a way. But they are realistic depictions, but not the real thing at all.
So in the Disney version, they'll be singing viruses and they'll all be covered. I get it. Okay. Well, then moving on. Oh, sorry. I got to ask you this about size. When you say they're small, give us a sense of the scale. How small are we talking about with a virus? I mean, you can't see with a optical microscope, but how small? Can you put in a scale we can fathom?
Can I'll give you a sort of rough analogy, and of course analogies are never perfect, but if one of your cells is a basketball court,
I love examples like this. Yeah, go ahead.
Virus would be a basketball.
Wait. One of your cells is a basketball court. The virus is a basketball. Wow.
Yeah.
Cool. I can see that. Thank you. I love examples like that. All right, from George Jacobs. What do you know about the several dozen autopsies done in Italy revealing that death was caused by blood clots, not pneumonia? He says the real problem appears to be cardiovascular and not respiratory. Is this why, he asks, such a large number of patients never survive the ventilators? What do we say to George?
Well, not being a clinician, it's hard to really answer that question, but I totally recommend that he tune into the This Week in Virology podcast, where they're fantastic about answering emails that people send in. The podcast lasts like 2 hours because they spend a huge amount of time answering emails. But yeah, so it seems like this virus is affecting many different parts of the body, and for some people, that ends in the lungs. And for others, then it moves on to thrombosis, these clotting issues. So to what extent do ventilators play in this equation? I mean, it's hard to say, right? For not everybody ends up having these clotting issues. So ventilators may be great in some circumstances, but another issue with ventilators is that sometimes patients are coming into the hospital too late. The disease has progressed so far, and they end up on a vent, and it's really difficult to get their immune system back down to where it's not causing such devastation to the lungs. So I really don't have an answer to his question. There's so much to be learned about that, but there clearly is a second or third stage to the disease that involves clotting issues.
All right, well, we're going to move along because we have a bunch more questions that have just come in. So we'll try to be close. We're running out of time. Pam Cray wants to know about the relationships between different sciences, virologists, epidemiologists, public health concerns, officials, medical practitioners. Maybe, I don't know, it's a lot there, but what's the difference between a virologist and epidemiologist? Maybe we could talk about that briefly. Or not.
I can try to at least. Well, let's see. And Maureen, you can jump in at any point. So a virologist could be looking at really any aspect of the virus. So it could be someone who's, really looking at studies of the structure of the virus or the details of the replication of the virus inside the cell, or how the immune system responds to the virus. And whereas an epidemiologist is really primarily concerned with the transmission of disease, and certainly not all epidemiology has to do with infectious disease, but also, what are the patterns, environmental exposures, um, um, social factors in disease, all of those factors that explain the pattern and distribution of disease.
All right. I'm sorry, get a short, just we got a big backlog, so we're going to move on, but thank you.
Yeah.
Sorry to be the bad guy. Okay, I'm going to just, some of these we've answered, so I'm just going to acknowledge how do pandemics end from Kathy Maloney. I think we've touched on that. Deb DiPietro, we've mentioned that, her question. Gina Pilius, I think we've also touched on this. She's hearing on the news that people who have tested positive for the coronavirus are now testing negative for the antibodies for it. Is that possible? How could that be? You want to?
Sure. Well, hi, Gina. And as I mentioned, some of these tests have less than 100% accuracy. And so a small number of people who actually have antibodies have been exposed to the virus and made the antibodies, the test will come up negative just because of that low level of inaccuracy.
Okay, we have this is from, oh God, I hope I'm pronouncing this right, Laiba Zulfikar. How does the virus affect the lungs? What was originally the source of the virus? We've touched on that, I think. I heard the virus was caused by eating bats. Though I'm not sure, is that true? And if it is, then what germ is in the bat which causes the disease in humans? So
unlikely that it was from eating bats, but simply, we tend to share habitats with bats, and there's a lot of them. There's so many different bat species, and so I think it's more the case of bats cohabiting in the same spaces, and we're exposed to their excretions, and bats have a lot of different viruses that will circulate among bats and not necessarily cause disease in the bat, but then if it does spill over into humans, then it can cause disease in humans.
And it should be said that bats, in the terms of the larger scheme of things, bats do a lot of good in the world. They eat a lot of insects, among other things. From Dan Freeman, would you discuss why some viruses you can develop immunity to and others, like the common cold, you can't? I think we've touched on that. Thank you, Dan. You foresaw that question. From Joanne Meyer, there's been a lot of discussion about the amount of time spent in the presence of a quantity of virus. For example, if one wears a typical mask but is exposed to X number of viruses in 15 minutes in a large grocery store with some other semi-careful shoppers, what is the critical number of viruses that could take hold and eventually replicate dangerously? If there are a few inhaled, can the immune system clean them up, or are the few that get inhaled just a slower time bomb? Ooh, okay. In other words, yeah, that sort of reminds me of the allergy question. You could have one little thing that you react to and boom, you start sneezing. Is it like that?
That's a good question, and we don't know that number. And so probably best to assume it's low so that we continue with precautions that keep our exposure as low as possible. But she's right. There is sort of a probably a critical number that if you think you need to be exposed enough to start an infection, and one of the like many things in biology, the virus's strategy is make lots and lots of progeny, and only some are going to survive.
Mm.
And that's the case with viruses. Tons and tons of viral particles expelled, but only a few are actually going to, by luck, end up in the next host. You know, it'll end up on the wrong cell. It'll end up on your skin instead of in your lungs, and it won't be able to start an infection.
So this sort of presages the next question, because but first I want to ask you about, because it relates, numbers. I mean, when we talk about viruses, we're talking about millions, trillions. I mean, the numbers are astonishing, right? They're mind-boggling. I mean, how many people are on the planet? We're talking the population of planet Earth now is what? Anybody know? I don't, sorry. But viruses outnumber us exponentially.
They are the most numerous. I wouldn't, maybe we can't say living things, but the most numerous biological entities on the planet, for
Okay,
sure.
so keeping that in mind. So the next question is from, we don't know. Oh, no, we don't know. This is anonymous. If the virus, quote, comes from animals, why don't viruses come from humans?
Well, I think they do also come from humans, but the ones that are new to humans have to come from some other place than humans. So it's really the new viruses we see come from from animals,
Okay.
non-human animals.
Okay, so you can blame the thinning of the ozone layer on us, but not this. It seems this pandemic will never come to an end until there is not even one infected person. Is this so? From Pam Cray. That's our last question, I think. And it's talking about ever coming to an end. We've sort of answered it, but since we're at the end, let's answer it again. Is that true?
As long as other animals can't get infected, and there's some evidence that's dogs can be infected by this coronavirus. Whether or not they can transmit it back to humans, we don't know. But the presence of an animal reservoir is really important, because you could end all human to human transmission, but if it's in some other animal, then it's going to come back, potentially.
Okay. Oh, we have word that earlier question was from Julio Esteban. Thank you, Julio. And thank everyone who wrote in. I think we got to them all. And my deep thanks to both of you. I'd like to just say, I don't want to, Pam's going to close up, but I want to say that you both volunteered very generously to continue this discussion online, either on the Rosendale site or on the Facebook site. If people want to continue asking questions, if we didn't get something comes up, for a couple of days from now, and you want to post a question online, Maureen and David have very kindly said that they will answer it eventually on those places. So Pam, if you want to weigh in, my thanks to both scientists, and thank you all for listening.
Thank you.
I hope everybody out there, thank you all for coming to tonight's event. I've been sitting here riveted. I find the conversation and discussion has been riveting for me to listen to. The pieces of the puzzle come a little bit more into view, and I think that maybe every little bit helps. We did have a few questions about finding this discussion afterwards. You'll be able to find the link to get the recorded discussion on our website, the Rosendale Theater website. I want to thank you three for the generosity of your time and answering all these questions and discussing this. If everybody out there doesn't want to become a microbiologist at this point and study viruses, if not the whole microscopic, only found by DNA world, I don't know. I'm fascinated, and I'm so happy to be here. I also want to, so let me just thank you all. Maureen, Dr. Maureen Morrow, Dr. David Esteban, Dr. Jim Metzner.
Yeah, doctor, I'll take it. It's all right.
And I'd like to say a few thanks to people behind the scenes here. First, Justin Peony, our theater tech director, for switching gears from doing all the tech in the theater to getting this together so that we can live stream. And Anne Citron, our theater director, our amazing volunteer effort that continues to drive the Rosendale Theater, the whole board. I'd also like to thank Wendy Saul, who was instrumental in getting this panel together, as she has gotten all the Science on Screen panels together and has put together a science advisory committee for the Rosendale Theater to work on events that combine film and science and other kind of live science-oriented events. So thank you, Wendy. And I'd like to thank the Science on Screen program. We have become part of this through the grant from the Coolidge Corner Theater Foundation with major funding from the Sloan Foundation, the Alfred P. Sloan Foundation. If you haven't heard of the Sloan Foundation, they fund a lot of science education and are in this now hybrid area of how to relate entertainment to science education, and we're all in. So tonight's discussion and last week's discussion on death and dying can be found on our website after tonight. Next Tuesday, we have another Science on Screen discussion at 7 p.m. on space travel and exploration that was going to be paired with 2001: A Space Odyssey. We are going to show the movie in the theater on the big screen with the big sound, but in the meantime, we are going to continue our discussions online so that we don't drop out. We have many things going on here at the Rosendale Theater online and ready to come back in the theater. As you can see, I'm here and it's empty and I got a little cold because of it, but okay, that's it. I just want to say thank you. I'm Pam Cray. Thank you very much, everybody, and good night.
Thank you.
Thank you, Pam.